In the last lecture of this module, we will introduce two more helpful concepts. The first is the Lewis definition of acids and bases, which we've mentioned just briefly previously. This is an even more encompassing classification of acids and bases than the Bronsted-Lowry definition. We will introduce it here in the next few slides. You'll typically see the Lewis definition of acids and bases more fully employed in more advanced chemistry courses, such as introductory organic chemistry. But we'll spend a few minutes in this lecture outlining some of the basic concepts of this Lewis definition of acids and bases. The second thing we'll cover in the lecture is asking the question, what about the actual structure of a molecule, meaning the types and arrangements of atoms and bonds, makes that molecule a strong or a weak acid or base? We'll spend some time on both of these concepts before diving into the Lewis definition of acids and bases. I want to kind of whet your appetite concerning the looking at the actual structure of molecules and how that affects whether they're strong or weak acids or bases. Now, a molecule makes a great acid if it has an arrangement of atoms and bonds that allows it to easily donate H+ ions, because remember, we're mostly thinking in this course of acids as H+ donors. So what about a molecule, its structure, and the atoms that comprise the molecule would make it either better or worse at donating H+ ions? So as an example here, we'll use our well-known acid, acetic acid, and we'll look at the ionization of acetic acid using some 2D Lewis structures. So here's the Lewis structure of acetic acid. Here's the Ka value, which you know pretty well by now. And the hydrogen here is in red because we say that's the ionizable hydrogen. That's the hydrogen ion that will come off when acetic acid acts as an acid. So when that happens, you form the acetate ion. Here, the Lewis structure of the acetate ion is shown. And of course, you produce a hydrogen ion. Now, how easily H+ can leave this electron pair behind. You'll notice that once H+ leaves, it leaves that electron pair behind and a net negative charge here. How easily that H+ can leave will determine the strength of the acetic acid. One could ask the question, could we alter the structure of acetic acid to make it easier for the H+ to leave? In other words, could we alter its structure to make it a better acid? To do this, we would need to find a way to pull that shared electron pair away from the hydrogen ion in the OH bond and pull it toward the oxygen. So let's propose the following structure. Suppose we substituted for a hydrogen here a fluorine atom. And remember, fluorine is very electronegative. And I've drawn the fluorine without the lone pairs here, just to eliminate some crowding in the drawing. This is called fluoroacetic acid, and indeed, that minor structural change does make this a more powerful acid than acetic acid. You can see it's still considered a weak acid, but it's about 100 times stronger, according to its Ka value, than acetic acid. We say the inductive effect of the very electronegative fluorine atom pulls the shared pair, electron pair, closer to the oxygen atom in the OH bond, and now H+ can leave more easily. So here's fluorine, because it's very electronegative, pulling electron density toward itself, which has the net effect of pulling the shared pair closer to the oxygen, making it easier for H+ to leave. And now we get the formation of the fluoroacetate ion and a hydrogen ion. And again, as I mentioned previously, fluoroacetic acid is almost 100 times more acidic than acetic acid. A complementary and perhaps more important explanation for the increased acidity of fluoroacetic acid is that the electronegative fluorine atom stabilizes the fluoroacetate ion by effectively drawing the -1 charge left behind by the departing H+ away from the oxygen atom. That was kind of a mouthful. What am I saying here? Well, if you look at the fluoroacetate ion, it has a negative charge that's been thrust upon it since H+ left, and this ion has to figure out a way to stabilize the structure with this negative charge here, and the fact that the fluorine can draw electron density toward itself helps stabilize this structure because it pulls that single negative charge toward the fluorine. And as a result, the fluoroacetate ion is more stable than the acetate ion. The fluoroacetate can handle that negative charge better than the acetate ion can, and so the formation of fluoroacetate with the resulting ejection of an H+ is more favored for fluoroacetic acid. So that's a complementary or alternate explanation for why fluoroacetic acid is a better acid than acetic acid. So before going on more on the effect of molecular structure on the strength of acids or bases, let's go back to our initial point, the Lewis definition of acids and bases. Simply put, an acid, under the Lewis definition, is an electron pair acceptor, and a base is an electron pair donor. Now, the advantage of the Lewis definition, which seems oddly different than the Bronsted-Lowry definition, is that it expands the number of species that we can consider acids and bases. So the Lewis definition, and I'll show you this in just a second, encompasses all of the Bronsted-Lowry acids and bases, but allows us to expand our view of how many substances can act as acids or bases. So let me just show you an example. Let's examine the reaction between H+ ion and OH- ion in aqueous solution. Now we know that this is an acid-base reaction under the Bronsted-Lowry definition, but let's look at it with a different pair of eyes. So here's the hydrogen ion, here's the Lewis structure of the hydroxide ion, and we know that this forms water. Under the Lewis definition, H+ ion is considered an acid because it's an electron pair acceptor. Well, how is it acting as an electron pair acceptor? It's forming a covalent bond with oxygen by accepting this pair of electrons from oxygen to form a bond. So H+ is accepting this electron pair on hydroxide. So it's acting as an electron pair acceptor. And the hydroxide then is the electron pair donor, so it's called the Lewis base. Also, you will recall, of course, that H+ is a Bronsted acid because it's an H+ donor, and hydroxide is a Bronsted base because it's an H+ acceptor. So both the Lewis and the Bronsted-Lowry definitions consider H+ to be the acid and OH- the base, but for different reasons. Again, the advantage of the Lewis definition is that it expands the types of interactions we can label as acid-base reactions. To show you this, we'll recall an example from lecture 5 where we stated that aluminum 3+ ion dissolved in water is really a complex of aluminum with 6 water molecules attached by coordinate covalent bonds. And we wrote the formula like this, and we understood that the oxygen atoms of the water were attached to aluminum with these coordinate covalent bonds. Now, as we've done this before, let's redraw one of these aluminum-water interactions. So we have an aluminum ion, we have a water molecule, And I've highlighted one of the lone pairs on the water molecule, and I can actually draw an arrow here and say this lone pair is donated to the aluminum ion to form a coordinate covalent bond. Sometimes those are also called dative bonds. Anyway, both electrons in the shared pair originated from the oxygen atom in water. In any event, now we can see the aluminum 3+ ion acting as a Lewis acid because it's an electron pair acceptor, and the water molecule acting as a Lewis base, the electron pair donor. Aluminum 3+ is not considered an acid under the Bronsted definition. It is not acting as an H+ donor. And water here is not acting as an H+ acceptor, so it's not acting in a way that's consistent with the Bronsted definition of a base. So although this is not an acid-base reaction under the Bronsted definition, it is an acid-base reaction under the more comprehensive Lewis definition. So to summarize, the Lewis definition includes all the acids and bases under the Bronsted-Lowry classification, but allows for an expanded set of reactions to be considered acid-base interactions. Let's look at a few more examples of interactions that can be called acid-base reactions under the Lewis definition, but would not be adequately covered by the Bronsted-Lowry definition. In general chemistry labs, a common method for getting insoluble silver chloride to dissolve in water is to expose it to an aqueous solution of concentrated ammonia to form the soluble silver ammonia complex. What's happening here is that two ammonias molecules basically kick out the chloride from silver to form this silver ammonia complex and free chloride. Now we can write an equilibrium expression for this: silver chloride solid plus the two aqueous ammonia molecules forming the silver ammonia complex and free chloride. Now this equilibrium strongly favors the right-hand side, which is why it's such a handy way to get silver chloride to dissolve in solution, in an aqueous solution. If we draw the Lewis structures of the two ammonia molecules here and highlight the lone pair on the nitrogen, we can illustrate the fact that these lone pairs are acting as Lewis bases and donating these electron pairs to the silver ion, which is acting as a Lewis acid. As another example, nickel metal can be purified by isolation of something called tetracarbonyl nickel by reaction of carbon monoxide, which is acting as a Lewis base, with a nickel metal, which is acting as a Lewis acid. So we can illustrate this by writing nickel solid, and we can imagine this nickel solid as being part of an impure mix or ore, and we expose this to carbon monoxide gas under the right conditions. The carbon monoxide molecules will specifically go after the nickel metal and form what's called tetracarbonyl nickel, and this equilibrium also strongly favors the right-hand side. If we draw Lewis structures here to illustrate what's going on, I drew here the Lewis structure, an enlarged Lewis structure of a carbon monoxide molecule with the relevant lone pair on carbon highlighted, and that lone pair gets donated to a nickel atom and forms a nickel carbon monoxide bond. Carbon monoxide acting in this way with a metal takes on the name carbonyl, so this would be a nickel carbonyl bond. So CO here acts as a Lewis base. It's an electron pair donor. Nickel acts as a Lewis acid and accepts electron pairs from CO. And you can see that these interactions here would not be considered acid-base interactions under the Bronsted-Lowry definition. However, under the expanded Lewis definition, they are considered acid-base reactions. Let's go on to now talk about the second point of this lecture, which remember I elaborated on the first slide as an introduction. What about the actual structure of a molecule? The types and arrangements of atoms and bonds makes that molecule a strong or a weak acid or base. We will limit our discussion here to acids and bases that can be described in the Bronsted-Lowry sense. That is, what characteristics make a molecule a weak or strong H+ donor or acceptor. So after introducing you to the Lewis definition, which is an alternate definition, I just want to remind you, as I've told you before, that for most of this course, we will view acids and bases in the Bronsted-Lowry sense, and our discussion here of particularly the strength of acids will look at acids in the Bronsted-Lowry sense. So what we're talking about is what would make an acid a good or a not so good H+ donor. Now acids can be grouped into several categories. The first is carboxylic acids. These are acids which contain the carboxyl group, often written as COOH, of which acetic acid is a member. Here's our acetic acid. Also remember fluoroacetic acid has this carboxyl group, and something called benzoic acid has the carboxyl group. I have Lewis structures here for these three. Here's the familiar Lewis structure for acetic acid. We've already seen the structure of fluoroacetic acid. And just to remind you, I've omitted the lone pairs on the fluorine. And here's the Lewis structure of benzoic acid. This structure here, just to remind you, is a 6-carbon ring. So each vertex here is a carbon, and then there's one hydrogen off each carbon, and this ring is an alternate double and single bonds. It's what's called a aromatic ring. That's the C6H5 part of benzoic acid. And also just to remind you, these are Lewis structures, so I am not really showing the full extent of some of the geometry here. So my geometry around the carbon atom reflects what is probably there. The COH bond is probably bent, and sometimes I'm better or worse at showing that. In any event, carboxylic acids are often referred to as organic acids. A second type of acid that you commonly see in general chemistry courses are called oxyacids. Now these are acids which are composed of OH groups, and you think, well, that's odd. I thought that was a base. Well, hang on for a second. And or a mixture of OH and double bond O groups, all attached to a central atom. So here's some examples. Sulfuric acid, here's its Lewis structure. And you'll notice here it has 2 OH groups and 2 what are called oxo groups. That would be the double bond O. And of course, these are the ionizable hydrogens. And a weaker acid here would be chlorous acid, which contains 1 oxo group and 1 OH group. Finally, the last category of acids that we'll look at are called binary acids. These are acids composed of just 2 different elements. This doesn't mean there's only 2 atoms in the acid. It means there's just 2 different elements. So an example would be the commonly known hydrochloric acid, 2 different elements, hydrobromic acid, 2 different elements, hydrosulfuric acid, H2S, dissolved in water, 2 different elements. So there's 3 atoms, but only 2 different elements involved. And here are the Lewis structures of these 3. And for hydrosulfuric acid, for some reason, I decided to draw the Lewis structure that reflects the real bent geometry around the sulfur atom. Now let's briefly look at how we might rationalize the variation in acid strength within each of these categories of acids. In carboxylic acids, which contain this carboxyl group. We've shown in a previous slide that fluoroacetic acid is significantly more acidic than acetic acid, and attributed this to the inductive effect of the very electronegative fluorine atom in fluoroacetic acid on the shared pair of electrons between the oxygen atom and the hydrogen atom in the OH bond of the carboxyl group. So here we have acetic acid. Here we have fluoroacetic acid with the lone pairs omitted on fluorine. And you can see that fluoroacetic acid has a significantly larger Ka value than acetic acid. And we talked about the inductive effect of the fluorine pulling electron density toward itself, making this electron pair pull closer to the oxygen, making it easier for that hydrogen ion to leave. And recall also that the stability of the anion formed is an important factor. Carboxylic acids, just by the nature of their structure, have kind of a built-in method for dealing with the negative charge left behind by the loss of hydrogen ion. The negative charge can be delocalized between the oxygen atoms and the carboxylic acid group, and chemists like to demonstrate this by drawing resonance structures, which hopefully you saw in General Chemistry 1. In any event, this is an easier concept than what it just sounded like. Let's look at acetic acid. So here's a structure of the unionized acetic acid. If it does ionize, it spits out a hydrogen ion and produces an acetate ion. But you know, there's another way we can draw this. We can draw an equally valid Lewis structure by putting the negative charge on this oxygen in the double bond here. So these are resonance structures, two different possible structures, Lewis structures for the acetate ion. For fluoroacetic acid, we can draw a similar structures. So we have here the fluoroacetate ion, and that can also be drawn in two different forms, so there's two resonance structures. Okay, so what's the point? Well, acetic acid can act as an acid because the acetic acid, even when it loses the H+, does have the ability to deal with this negative charge by what we call delocalizing it over the carboxyl group. And we signify this delocalization by drawing these two resonance structures. Now, fluoroacetic acid has an even better chance at handling this negative charge, as we said before, because it too tries to delocalize the negative charge around the carboxyl group, but it can also, pull electron density away from this part of the molecule and stabilize this anion even more. So both acetate and fluoroacetate ions are stabilized by delocalization of the negative charge left behind by the departing hydrogen ion. We show that delocalization by drawing valid resonance structures. However, the fluoroacetate ion is more stabilized due to the inductive effect of the electronegative fluorine atom. Thus, fluoroacetic acid is more acidic than acetic acid. The next group of acids we want to discuss are called oxyacids, and we mentioned these are acids composed of OH groups and or a mixture of OH and double bond O or oxo groups all attached to a central atom, which usually for us is a nonmetal. And we find that the acidity of oxyacids increases as the number of oxo groups attached to the central atom increases, and the electronegativity of the central atom increases. Now, both of these can be viewed as making it easier for H+ to leave the molecule by polarizing the shared electron pair in the OH bond towards the oxygen atom, and by stabilizing the negative charge on the anion form by the departure of the H+. Let's look at some examples. Let's look at the increasing acidity as the number of oxo groups attached to the central atom increases. We're going to look at a series of related oxo acids or oxyacids. This is hypochlorous acid. This is chlorous acid. This is chloric acid. And this is perchloric acid. Hypochlorous acid is very weak. Chlorous acid is one of the stronger weak acids. Chloric acid is considered a strong acid, and perchloric acid is considered a very strong acid. So what's changing as we go through this series? Well, you can see the number of oxo groups is increasing. So hypochlorous acid has no oxo groups. Chlorous acid has 1, chloric acid has 2, and the perchloric acid has 3. So the acid strength is increasing, going from left to right. And what's going on here, again, is that these oxo groups are pulling or polarizing the OH bond here, pulling the electron pair closer to the oxygen, making it easier for hydrogen ion to leave. There's something else that's curious about this series. If you actually assigned oxidation numbers to the chlorine in each one of these, you'd find out that it's +1 in hypochlorous acid, it's +3 in chlorous acid, it's +5 in chloric acid, and it's +7 in perchloric acid. Therefore, we might also correctly say that the acidity of oxo acids increases as the oxidation number of the central atom increases. Now, the second effect we talked about up here was the electronegativity of the central atom. We say oxyacids become more acidic as the electronegativity of the central atom increases. So let's look at an example here, or some examples. Let's look at hypoiodous acid and hypobromous acid and hypochlorous acid. Now, one of the things you might want to notice here is that the central atom in these structures appears to be oxygen, but by central atom here, we mean these changing attachments to the OH, iodine, bromine, and chlorine. You'll see that for hypoiodous acid, it's pretty weak. Hypobromous acid is a little stronger. Hypochlorous acid is a little stronger yet. So acid strength is increasing, going from left to right. The electronegativity of iodine is 2.5. The electronegativity of bromine is 2.8. And the electronegativity of chlorine is 3. So you can see as the electronegativity of the, quote, central atom increases, the acid strength increases. For the same reason as we've seen before, the increased electronegativity of these halogens that are attached pull that electron pair closer to oxygen, making it easier for H+ to leave, and would also stabilize the resulting anion better as the electronegativity of these three halogens increases. So let's look at our final category of acids that we listed previously, binary acids. Again, these are acids composed of just 2 different elements. Some of the most common binary acids discussed in general chemistry courses involve the aqueous solutions of HF, hydrofluoric acid, HCl, hydrochloric acid, HBr, hydrobromic acid, and HI, hydroiodic acid. All of these acids involve hydrogen attached to a group 17 element, in other words, attached to a halogen. So here's a space-filling model, the best I could do, of hydrofluoric acid. It's, of course, considered a weak acid. And remember, a weak acid is just an acid that does not ionize appreciably in water. The term weak does not mean safe. So, for example, hydrofluoric acid causes severe burns and will dissolve glass. It's one reason why you'll never see it in a general chemistry lab. The more familiar hydrochloric acid is considered a strong acid. The even stronger hydrobromic acid, of course, is also considered a strong acid. And then there's hydroiodic acid, which is also considered a strong acid. Now in water, it may be hard to distinguish between the strength of hydrochloric, hydrobromic, and hydroiodic, because all of these would simply be leveled to the hydronium ion. However, it can be shown under other conditions, that would mean in non-aqueous solutions, that HI is a stronger acid than HBr, and HBr is a stronger acid than HCl. Well, how can we explain this trend in acidities? And the trend here, if it's not obvious by now, is that hydrofluoric acid is weak, hydrochloric acid is stronger, it's strong, and then as you go down, the acids become stronger and stronger. We're going to try to rationalize this in kind of an odd way. What I have here are gas phase bond lengths and bond dissociation enthalpies. And I drew these molecules like this intentionally to show you, or to try to reflect reality. So what do we mean by the gas phase bond lengths? What we mean is that the center to center distance between these two atoms, which would be the length of this dotted line here in an individual molecule in the gas phase, is 91.7 picometers. The bond length in HCl, which I'm trying to show here, is longer than in HF. It's 127.4, HBr even longer, and HI the longest. Now we're going to list the bond dissociation enthalpies, which is not the energy needed to separate an H plus from F minus. It's actually just the energy needed to break this bond evenly, the HF bond. In any event, if we list these, not surprisingly, the HF bond is the most difficult to break. It takes the most energy because it's the shortest. And then the bond dissociation enthalpies become smaller and smaller going down the series here. And so what's normally said in general chemistry textbook is that the acid strength is increasing, which it is, as the bond dissociation energies drop and the bond lengths increase. The order of acidities, HI is the strongest, followed by HBr, then HCl, then HF. So again, the typical explanation for the observed order of acidities for the series of hydrohalic acids is that acidity increases as bond dissociation enthalpies decrease and bond lengths increase. Now, interestingly, the electronegativity of each halogen atom in the series does not determine the order of acidities. Otherwise, HF would be the strongest acid. One more example of a binary acid. This is one we mentioned before. A very common binary acid that's discussed sometimes in general chemistry lectures and sometimes used in general chemistry labs is hydrosulfuric acid, which is simply an aqueous solution of hydrogen sulfide gas, which we can write as H2S aqueous. Hydrosulfuric acid is actually a weak diprotic acid, so we can write two ionization equilibria. The first would be where the H2S loses one H plus, and the Ka value for this is quite small. The second ionization would be where the HS minus resulting from the first equilibria can undergo further ionization, donate another H plus to form a sulfide ion and another hydronium ion. The Ka2 here is pretty small. We can actually write an overall reaction here, which would be H2S interacting with two water molecules forming sulfide and two hydronium ions. The overall K here is pretty darn small, 9.5 times 10 to the minus 22. Now, aqueous solutions of H2S it can be pretty foul-smelling if you get your nose near them, because it's possible for some H2S gas to outgas back out of the solution. And you want to be careful about smelling H2S gas. It's also called swamp gas. That sounds like it should be harmless. However, interestingly enough, it's actually quite toxic. So an interesting use of H2S solution, in other words, hydrosulfuric acid solutions, in a lab would be to regulate the concentration of sulfide ion in an aqueous solution through adjustments in the pH. Here's what we mean by that. If you look at either the overall equilibria or either one of these individual equilibria, but we'll look at the overall one, I guess. If you look at those and remember Le Chatelier's principle, you can see that if you intentionally add hydronium ions to the solution, in other words, add acid, you're going to push this equilibria back toward the left, and that's going to lower the concentration of sulfide ions in the solution. Conversely, if you wanted to maximize the concentration of sulfide ions in solution, you could add a base, you could add some hydroxide to react with the hydronium ion, raising the pH and pulling this equilibrium to the right, thus producing more sulfide ions. So if you needed sulfide ions for a particular reason, and you needed to control or change in a subtle way the concentration of sulfide ions available during an experiment, you can do that by simply changing the pH of the solution. At low pH, fewer sulfide ions. At high pH, more sulfide ions, depending on which way, of course, you're pulling this equilibrium. In part 2 of this lecture, we will go on to consider weak bases and the effect of molecular structure on the basicity of a certain class of bases called amines.